All right, welcome to This Explainer. Today we're diving into something that sounds small, but is literally a matter of life and death in fisheries management. We often think of fisheries as just managing the fish themselves, right? But today we're zooming way in. We are exploring the absolute foundation of the aquatic trophic pyramid, primary producers. Understanding their complex cellular architecture isn't just some academic exercise, it's an active, on-the-ground diagnostic tool that is absolutely crucial to preventing total ecological collapse. Let's get into it. Okay, picture this. You are a provincial fisheries officer facing what we'll call the Pangasinan milkfish crisis. You're standing in front of 5 earthen, brackish water nursery ponds, and overnight they have suddenly turned a bright, toxic-looking green. The pond operator is absolutely panicking. They're terrified of an immediate crop failure, and they want to spray heavy, synthetic, copper-based algicides right the second to clear the water. So, to spray or not to spray? Your decision in this exact moment will dictate the fate of that entire ecosystem. No pressure, right? Let's unpack the stakes here in Chapter 1, the Pangasinan Milkfish Crisis. Here is the massive dilemma. Acting without doing your biological diagnostics first could trigger a catastrophic disaster. If you blindly spray a beneficial green microalgae bloom, you'll instantly trigger a lethal oxygen crash. Millions of algal cells will die, and the aerobic bacteria that decompose them will completely exhaust the remaining dissolved oxygen in the water within mere hours. The milkfish will suffocate. But on the flip side, what if that green mock is actually a toxic dinoflagellate bloom? If you hit that with a copper-based algicide, you'll rupture their cell membranes and dump massive amounts of deadly paralytic shellfish toxins straight into the water column. The milkfish absorb it, and they become totally lethal for human consumption. Basically, a wrong guess here is disastrous. Which brings us to Chapter 2, Identifying Microscopic Suspects. Before you even think about touching those chemicals, our microscopic detective, that's you, needs to prepare a wet mount slide and get that water under a compound microscope. We are basically looking for 3 main suspects. First, beneficial diatoms. You can spot them by their glassy, perfectly symmetrical silica cell walls. Second, beneficial chlorophytes, which are just simple, non-toxic green cells. And third, the really dangerous ones, toxic dinoflagellates. These guys show off thick, armored cellulosic plates and very distinct flagellar grooves. Oh, and here's a super cool lab trick you have to use here. You must place a tiny droplet of petroleum jelly at the corners of your cover slip to act as a spacer. Unicellular flagellates are incredibly fragile, and without that jelly giving them a little room, you'll literally crush their natural 3D flagellated structures before you can even identify them. Moving right along to Chapter 3, Prokaryote versus Eukaryote cells. If you're going to manage these ecosystems properly, and this is actually a core competency for officers under the PQF Level 6 framework, you absolutely have to be able to distinguish the cellular architecture of these primary producers. It's a huge evolutionary divide. On one side, you have prokaryotes, like cyanobacteria. They completely lack a nucleus or any membrane-bound organelles. Their genetic material is just loose in there, and their thylakoids float freely. Then, on the other side, we have eukaryotes, like our diatoms and green algae. These feature DNA neatly organized within a double membrane nucleus, specialized compartmentalized organelles, and highly structured chloroplasts. Oh, and they also handle buoyancy in completely different ways, which is wild. So, prokaryotes, like cyanobacteria, don't have vacuoles. How on earth do they float? Well, they use something called gas vesicles. These are rigid, hollow, protein-walled cylinders inside the cell. They are totally permeable to ambient gases, but strictly impermeable to water. By inflating or collapsing these little vesicles, cyanobacteria act like tiny microscopic submarines. They float up to the surface on overcast days to catch the light, or they sink down when the midday sun gets too intense. And this vertical migration is exactly what creates those thick, toxic surface scums that block light from reaching the producers down below. Then, on the good side of a spectrum, we have diatoms, basically the gold standard for aquaculture feed. They build themselves this incredible evolutionary energy-saving glass house known as a frustule. So instead of standard cellulose, this cell wall is made entirely of polymerized silica. Why does that matter? Because synthesizing silica takes significantly less cellular energy, less ATP, than building complex organic cellulose polymers. When there's a nutrient upwelling, diatoms can reproduce exponentially and just outcompete other algae, simply because their houses are practically biologically free to build. All right, let's dive into Chapter 4, Extreme Marine Cellular Adaptations. These plant and algal cells possess some truly incredible biological mechanisms to survive highly stressful, hypersaline, and light-limited marine ecosystems. Imagine an earthen, brackish pond evaporating under the sun and hitting a hypertonic salinity of 42 parts per thousand. That creates a severely hypertonic environment, which naturally wants to suck the water entirely out of the plant cells. This intense osmotic stress draws the water right out, causing a lethal shrinking known as plasmolysis. This is where the cytoplasm literally pulls away from the cell wall. Yikes. If a fisheries officer observes this under the microscope, they have to act fast. They must flush the pond with fresh water immediately to drop the environmental osmolarity and get that turgor pressure restored. But estuarine plants aren't going down without a fight. They have a natural defense mechanism against this osmotic stress. Their central vacuole is bound by a specialized active transport membrane called the tonoplast. The tonoplast actively pumps sodium and chloride ions inward, which keeps the intracellular solute concentration incredibly high. Basically, by making sure the inside of the cell is saltier than the seawater outside, water is naturally drawn back into the cell. This preserves the turgor pressure needed to keep the plant alive and upright. It's a brilliant biological pump. Now let's completely switch gears and look at another extreme: darkness. Down at depths of 150 meters in the ocean, standard terrestrial green chloroplasts are completely useless. Why? Because red and orange light wavelengths are entirely absorbed in just the first 10 meters of water. Only blue and green light penetrate this deep. So how do deep-water red algae survive this near darkness? They utilize a genius accessory pigment called phycoerythrin. This specific pigment absorbs those deep penetrating blue-green light wavelengths and then transfers that captured energy directly over to chlorophyll A. It's an absolutely spectacular evolutionary workaround that lets red seaweeds happily photosynthesize way down in the depths, where green algae would literally starve to death. Finally, let's look at Chapter 5, the biologist's optical toolkit. To actually see all these crazy mechanisms, whether you're hunting a microscopic killer dinoflagellate or just trying to resolve the tiny, intricate pores on a diatom frustule, you need perfectly calibrated optical instruments. To see those super fine details clearly, without the light bending and scattering everywhere, you absolutely must utilize high numerical aperture, or NA, objectives. When you're up at 100x magnification, placing a tiny drop of immersion oil, which perfectly matches the refractive index of glass, prevents that light scatter. Plus, you have to focus the light beam directly onto the specimen by raising the condenser and adjusting the iris diaphragm. That's the secret to achieving optimal resolving power. But just seeing the cell, not enough. We have to measure it so we can calculate biomass. Since ocular micrometer divisions inside the eyepiece are just completely arbitrary lines, you have to mathematically calibrate your lens against a known stage micrometer before you measure any pathogens. It's basically a four-step process. You align the two micrometers, count exactly where the divisions perfectly overlap, calculate your factor, and then apply it to your specimen. Let's look at how the math actually works. Okay, here's the optical math in action. Imagine a standard stage micrometer where each division is exactly 10 micrometers. If 12 of your arbitrary ocular units perfectly match up with 3 stage units, that means those 12 ocular units equal exactly 30 micrometers. You just divide 30 by 12, and boom, your calibration factor is exactly 2.5 micrometers per ocular division. So why does this actually matter in the field? Well, if you spot a suspicious, potentially toxic dinoflagellate on your slide and you see it spans exactly 16 ocular divisions, you instantly multiply that by your 2.5 factor. Now you know for a fact its exact true size is 40 micrometers. This exact precision is what allows fisheries officers to accurately calculate the cellular biomass and determine the exact carrying capacity of the entire pond system. We've really seen how identifying these invisible microscopic structures, from energy-saving silica frustules to the flagellar grooves of a toxic threat, is the ultimate defense against economic and ecological collapse in our aquaculture systems. By mastering the fundamental botany of primary productivity and utilizing high-precision optical diagnostics, fisheries managers are literally the frontline defenders of our waters. But all of this leaves us with a pretty provocative question to ponder. As our marine environments become more extreme and as we push the absolute boundaries of global aquaculture, how will our microscopic diagnostic tools need to evolve to protect the delicate trophic pyramids of tomorrow's food supply? Think about it. Thanks so much for exploring the microscopic world with me today. See you next time.